Bismillahirrahmanirrahim. Assalamu alaikum. Today we are going to start gametogenesis. Before starting gametogenesis, it's necessary to understand meiosis, which occurs in the process of gametogenesis. Gametogenesis involves meiosis. Meiosis is a division of cells which is called reduction division. During meiosis, number of chromosomes in the daughter cells decreases by 1/2, so the cells, that is the gametes formed, are haploid, means they are developed from diploid cells, but themselves they are haploid. So this process of reduction division is called meiosis. Meiosis is divided into two phases. One is meiosis 1 and second is meiosis 2. Meiosis 1 is actually a true reduction division because during meiosis 1 number of chromosomes are halved. So let's see. This is the starting of the meiosis, in which the chromosomes which were present in the form of chromatin is now being paired, and the process of pairing occurred in the phase called prophase. So this is prophase. In this phase the pairing of chromosomes occur. After this pairing of chromosomes are now paired. They have formed paired. Let's see their individual structures. This is the pair of chromosome, and this pair of this is pair of chromatids. This one is chromatid and these two are two chromatids of a chromosome. So this forms a pair with homologous chromosomes. This is pair of chromosomes which are further composed up of a chromosome, and one chromosome is made from the two chromatids. It is like this. This the process of their pairing, from which they form pair and unite. This is called centrosome. This is the point where two chromatids of a chromosome unite, is called centromere. This is centromere. This is chromatid. This whole is one chromosome. This is chromatid. This is called sister chromatid of the other chromatid of the same chromosome. These two are sister chromatids, but the chromatid of one chromosome compared to the other, these two are called non-sister. These are non-sister chromatids, and these two are sister chromatids. Fine. So this was a pair of homologous chromosomes, two chromosomes of the same type. So in the prophase, pairing of homologous chromosomes occur, and then it moves towards this. This is the phase called metaphase. This is metaphase. In the metaphase, two homologous chromosomes forming pairs are arranged in the center, and the spindles are formed from the two centrioles. These are the mitotic apparatus, and the spindle forms from the mitotic apparatus and holds the homologous chromosome pair individually. Each chromosome is being held from the centromere. This is the point called centromere. From centromere, the meiotic spindles grab the homologous chromosome, and the chromosome, they try to pull it towards the corners, towards the poles, right? Next, this is forming the central arrangement of homologous chromosome in the whole cell. If we take the whole cell, we are just having two homologous pairs of chromosome for our better understanding. Now the mitotic spindles pull the homologous chromosomes, individual chromosome of the homologous chromosome pair, and they try to move it apart. The phase in which the homologous chromosomes are separated is called anaphase. This is the anaphase. And now when they try to pull the chromosomes towards the poles, the homologous chromosomes are pulled towards the poles, and the cytoplasm start division, and there is a constriction, and in this way the whole cell is divided into two. Okay, then the whole cell divides into two. These are the result of these are the two daughter chromatids, but these are two daughter cells. This phase in which the cytoplasm now constrict and the cell is divided, cytoplasm and also nucleoplasm constrict and the cell is starting to divide into two individuals, is called telophase. Two individual cells. This was the meiosis 1. Meiosis 1 is completing here. Now from these cells, one we will take one for our understanding. I want to clear one thing here. You are observing there are some segments which are actually not from the same chromosome. Actually they belong to this chromosome. What is this? Actually in meiosis 1 there occur a process called crossing over. In crossing over, chromosomes which are present in the form of pairs, their parts are exchanged, and part of segments of one chromosome become the part of the other chromosome. This process is called crossing over. It's very important for the genetic recombinations. Fine. Now we are moving towards meiosis 2. Meiosis 2, take this cell here. Now we are seeing that these two chromosomes which were present, now only chromosomes are present, not the pair of homologous chromosomes are present. These chromosomes arrange in the form of line, and this is the linear arrangement. Then it is also called metaphase, and this time it is called metaphase 2 because it belongs to meiosis 2. It is meiosis 2. This was meiosis 1. Okay, this whole process is meiosis 1. Now metaphase arrangement in the metaphase give rise to the next phase, which is called anaphase. Keep in mind here there was the separation of homologous chromosome pair, and each chromosome of a homologous pair moves apart. Now there are not any pairs, any type of pairs present. So now the one chromosome give chromatids to separate. One chromatid move towards the other pole and the one towards the other pole. So this is anaphase, and then telophase occurs, the separation of the cytoplasm and the nucleoplasm, and finally two daughter cells are formed. So this was the whole process of meiosis. In meiosis, haploid gametes are formed from diploid cells. These were the 2n cells, means these were diploid cells, and the daughter cells formed after meiosis 2 complete meiosis are not diploid, but these are haploid, having only one set of chromosomes and called haploid. These are having two set of chromosomes and called diploid. Fine. So this was the meiosis. Now we will start spermatogenesis.